So, hello everyone. I'm Sofia. I'm final year PhD student in Wilfrid Herity's group at the Elam Institute. And today I'll tell you about the role of SNORD116, which is a Prader-Willi syndrome risk gene in cardiomyocyte differentiation. So, Prader-Willi syndrome is a complex developmental disorder, and it affects many systems, including neurologic, endocrine, and metabolic systems. And while much of the research so far has focused on the neural tissues, individuals with Prader-Willi syndrome also show a high incidence of cardiac abnormalities. And these heart issues have typically been associated with other dysfunctions, such as endocrine dysfunction, like growth hormone deficiency. But emerging evidence suggests that these cardiac symptoms are actually congenital, not simply consequence of those other factors. And SNORD116 is a cluster of CD-box SNORNAs that were described to you in the first talk of this session. And it's hosted in introns of a small nuclear RNA host gene 14, and is the critical region deleted or non-functional in most PWS cases. But its molecular targets remain unclear. So in this study, we set out to explore the molecular role of SNORD116 during cardiac differentiation. To do this, we used human-induced pluripotent stem cells that were differentiated into cardiomyocytes. And we know from prior research that during normal cardiomyocyte differentiation, SNORD116 expression gradually decreases. And we also know that a knockout of SNORD116 results in delayed cell proliferation, delayed cell growth, and reduced metabolic activity. So what's still unclear is why. We thought, could these phenotypes stem from disruptions to post-transcriptional RNA processing, such as splicing or free prime end formation? So for this, we used Oxford Nanopore long-read sequencing, which gives us full-length transcript information, making it ideal for examining differential transcript expression and usage and alternative polydinylation. And we had samples taken on days 2, 6, and 30 of cardiomyocyte differentiation from both wild type and knockout lines with 3 biological replicates per condition. So for us to be able to assess these isoform level changes, we developed a custom end-to-end pipeline that integrates splicing and alternative polydinylation analysis from long-read data, since, to my knowledge, no existing tool handled all of those components effectively. And so briefly, what I did was, so alignments were clustered into isoforms using isoquant, and then transcript models were refined using scanty3, and then the transcript start and end sites were validated against RefTSS and polyaside databases. And then for quantification, I used long-read Callisto, and the entire pipeline, including SNOGLOBE, SNORNA target prediction, and alternative polydinylation detection, will soon be available as a workflow on GitHub. So, interestingly, most of the differential transcript expression that we detected was at day 6, so the early cardiomyocyte stage, and that was over 1,200 isoforms showing a log-fold change of bigger than 1.5. But what's striking here is that between 19 to 31% of differentially expressed transcripts across these 3 time points weren't annotated in gene code, meaning they would have been entirely missed if we didn't build a sample-specific transcript annotation. So the first step of our analysis, the isoform switching analysis, pointed to dynamic 3' UTR regulation as a potential mechanism. And what we know is that, in general, longer 3' UTRs are associated with reduced mRNA stability, often due to increased microRNA binding. And the one enriched result that I found was that at day 2, I saw a significant enrichment for longer 3' UTRs. And although that effect wasn't significant, but you can see that by day 30, this effect was kind of reversed. So to validate our observation of 3' UTR dynamics, we performed a coverage-based transcriptome-wide analysis of alternative polydinylation, and using DAPARS2 long-read, which detects proximal and distal polydinylation sites, we confirmed a significant shift towards these distal further away poly-A site usage at day 2. And then finally, we decided to explore whether these transcripts of genes that were either differentially expressed or alternatively polydenylated might be direct targets of SNORD116. And we used a random SNORD96A and then also SNORD115, which is a related but distinct SNORRNA cluster that is also hosted in the Prader-Willi locus. We used those two as controls. So using SNOGLOBE, we predicted SNORRNA binding sites across the transcriptome and found that across 3 SNORD116 subgroups, subgroup 3 down here, have the most predicted binding sites. But interestingly, dysregulated transcripts were significantly enriched for both SNORD116, but also our control SNORD115. So this raises the possibility of potential coordinated regulatory role. But importantly, just to note, the overlap, there was barely any overlap between SNORD115 and SNORD116 targets. We also observed one interesting effect, that the enrichment of SNORD binding sites shifted towards the 3' UTRs in the alternatively polydenylated transcripts. So in summary, our data reveals a stage-specific post-transcriptional effect of SNORD116 loss during cardiomyocyte differentiation. We observed extensive isoform dysregulation, particularly at day 6, including many transcripts that weren't present in gen code. And we also saw these dynamic stage-specific 3' UTR changes with longer 3' UTRs at day 2 and followed by 3' UTR shortening at later stages. And we also found significant enrichment for SNORD116, but also SNORD115 binding sites among the affected transcripts. So in conclusion, I just want to add again that custom annotation is critical for capturing disease-relevant isoforms. And although we detected many potential targets, for us to distinguish between direct and indirect targets of SNORD116 would need lots of experimental validation, such as mutagenesis of SNORRNA binding sites, knockdown of target mRNAs. So yeah, and just at the end, I'd like to thank our collaborators, Terry Holmes, who basically generated most of the data that I analyzed for this project. And I'll also like to thank my funders.
Questions?
Of the different families of SNORD116, are they differentially expressed at the different time points that you were looking at?
So there were slight changes in coverage, but there was one issue, I'd say issue with my pipeline is the fact that it's tailored to mRNAs. So a lot of the kind of single exon, which RNAs are, a lot of the single exon transcripts are filtered out. So by the time I'm doing kind of the Callisto analysis, I'm not actually looking at SNORRNAs, but just based on coverage, yes, there is a difference in the different SNORRNA groups.
Okay, so if there are no further questions, then we'll thank the speaker and move on.